From Microbe TV, this is Office Hours for Wednesday, June 11th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Welcome to all of you tonight, and welcome to our mods. Please tell us where you're from so I can shout you out. We have some interesting items for you tonight. So moderating tonight is Les, Tom, Barb Mac, Vanity Nutrition, and Steph SF. Full house tonight. Oh boy, this is great. So again, everyone, shout out where you're from. Tonight's agenda, we're going to have some news items because, every week there's news going on about science and public health being screwed up in the U.S., so we're going to continue talking about that. And then we have a mini lecture, Evolution Part 2. This is a very cool part. You got all the nuts and bolts last time, so this time we are going to talk about some concepts and how viruses evolve. Okay. Let's see who's here tonight. Besides our intrepid moderators, of course. So Tom, of course, is from Oregon Coast Range, where it's 22°C. What is the weather here? It was a nice day today. In New York, New Jersey. It's currently 25°C and sunny out in the burbs here. Yeah, it's a good time to click like. If you click like, maybe we can attract more people to hear real science, right? Not the fake stuff, not the misinformation that comes from U.S. government officials these days. Wayne is in Urbana, Illinois, 85 or 29 and sunny. MN is in the D.C. area. Patricia is in Massachusetts. Leo is here from California. Truth or Dare is in La Jolla. Mark Martin, Tacoma, Washington, sunny and 20°C. That is microbial tomorrow, almost to 100. Celebrate those milestones, right? We celebrated on TWIV 100, 200, 300, 400, 500, and then 1,000. And that's it. No more numeric celebrations. TWIV 1000 was great in the theater, but what a hassle to organize. I didn't hire someone to organize it like an event planner, and I did it mostly myself, and that was just too much. Because those of you who went remember, after it, I just stood near the entrance to the reception area and looked stunned. Noir LeBlanc is in Albuquerque, the armpit of civilization. Oh my, 33°C. I've only flown to Albuquerque on the way to Taos. 3 hours to Taos. That's right. Steph SF in San Francisco, 61F. Feels much cooler due to wind and moisture, but was very foggy and overcast the first half of the day. But it's summertime, and that's great. Mike and Lori are in Seattle, 20°C. Eliza B. is in southern Ontario, where it is 26 and clear. Kang is in Chicago, 30°C with hazy skies. Mark Martin, mods. Wasn't that an expression for cool people in the 60s? If you were mod, yeah, you were cool. They're mod. Yep. Now it's in use for something else. Repurposing of the English language, right? Does that happen in other languages too? You repurpose words over time? John, yeah. Good to have you, John. Our favorite nephrologist. Carol from Bakersfield, where it's really hot, 36°C. So Leo's in Mendocino, 57F. Jefferson Space is in Miami. I just saw a billboard that went up in Miami, and I'm not going to mention it because we don't get political on this program. Maureen is in northeastern Ohio. John is in Minneapolis, where it's 20°C tonight. Tom told Harmit Malik, I prepped for a seminar with TWIV 205. He liked that. Looked up his wide-ranging work. Terrific stuff. Patricia, this is going to be an interesting podcast with a nightmare ahead for science. Folks, it just gets worse every day. Listen to Beyond the Noise. We cover it all over there. But Rob Basile is in Toronto. Welcome. Greg is in Palatine, Illinois. Welcome. Dion, hello again from the Bronx. Hope you're enjoying the summer. I wrote a few times. You're good. And the answer, Dion, is yes to your question. No problem. I will do it for you. I'm sorry I didn't respond immediately, but yes, I can do that for you. No problem. Laura is in Washington, PA, 26°C. Michael is in Mexico City, 21°C in the razy season. Elena Privyet from Reno, Nevada. Hello, Abdulaziz from KSA. Yes, we haven't seen you in a while. Glad to see you. I see you on Instagram, so I know you're around. So welcome back. Simon from San Jose. Have we looked at the new members of the vaccine committee? There's a New York Times article which talks about a couple of them. We'll go over that tonight. They're all horrible, right? They're just horrible. The people they replaced were seasoned professionals with no conflicts of interest, contrary to what RFK said. And these people are all anti-vaxxers. They're the ones who advise, the states about vaccines and vaccine schedules. Country's in for big problems, unless we can get rid of RFK Jr. Christina, Hershey, PA, it's 24°C. Sam likes the content. Welcome, Sam. Pete is in London. Hint of early summer. I've cut the front hedge and repaired 5 strips of rotten shiplap. Feeling smug. It's always good to get things done, isn't it? Logotic from Chicago, 32°C. Cheryl is in southern Ontario, 26°C. Veronika, dobry večer. I know that word. You taught me that a while ago. Privyet, Veronika in Moscow, 13°C. Good to see you again. Jill from Wilmington, Delaware, 78F. Yes, Logotic is looking up shiplap. I was going to ask, what is shiplap on a shed? Julie is in North Central Pennsylvania. Polka Dot Pup, Dennis in Texas, 71F and rain. Coralie from New South Wales, Australia. Jill can't wait for Matters Microbial 100, a huge accomplishment. We are very happy to propel all our podcasts forward. Crazy Lit Asin is in Bangkok, where it's 29°C. Noir LeBlanc, TWIV 1000 was phenomenal. We had a good time despite all the glitches, yes. John is in North Baltimore County, 50 miles northeast of Amy. And Veronika wants a shiplap. Be careful what you ask for, right? Cheryl is in sunny Santa Rosa, California, 71F. The Mod Squad. Yes, we have the Mod Squad here, for sure. Mods used to ride Vespas. Vespa, I remember the advertisement on television. They were at Vespa, papapa, 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 papayo di Vespa. Remember that? You could look it up. Doug is in Redondo Beach. We're at 66F and June gloom. Dogma. What a great name, Dogma. Bangalore, India, 5:30 a.m., 21°C. Thanks for joining us. Lolo's Lounge, it's L.A. Effing hot. Okay, so this shiplap is strip cladding with a slight overlapping tongue. Living River. Hello, Vincent. I was excited to hear one of my favorite podcasts reference Swift. What was that? Living River. Which podcast? I know it's not the Rogan, whatever it's called, but tell us what it is. Karen is from Southern California. Oh, Leo reminds us of the O2 Campy TV show Mod Squad. Yes, extremely campy. Soul Symphony. I'm doing my PhD in Mayo Clinic, and I wish I could come and shake your hands at ASV, but my lab can't send me this year. Still wanted to reach out. Thank you so much. I will be there this year doing a TWIV, of course. Looking forward to visiting Montreal for a few days. Sorry you can't make it. Okay, Living River says it's called If Books Could Kill, and they did an episode about the falseness of the lab leak. Good for them. I might check that out. You know, The Living River is the name of the book. I think it was by Edward Hopper. I know he's a painter. About his theory that, HIV came from polio vaccine trials, which is just wrong, but it's The Living River. Have you read it? Hello, Karen in Orange County, California. Serge is listening while doing the dishes in Ottawa. Excellent. Lauren is in a very smoky and 21 in Edmonton, Alberta. Okay, ASV 2026. I don't know where that is. Not sure. To look it up. New York Times published a list of 5 health-related sources one could trust, and Microbe TV was mentioned in the comments. That's great. Thank you for whoever mentioned it. I wish we were one of the 5 health-related sources, but I guess we don't have enough of a breadth of health-related stuff. But I'm happy to be in the comments. David is in super sunny Seattle, strangely. We got 4 S's there. S to the 4th power. That's great. Yes, Robert Malone. What? Oh my gosh. He's one of the members of the CDC vaccine committee. Appointed by RFK Jr. Joseph is in southwestern Ontario, Canada, where it's 28C. Bad Cat. Hello, Bad Cat. Winthrop, Maine, 68F. Truth or Dare asks the essential question. Would they be anti-vax if bitten by a rabid animal? No, they would not, because they're going to die if they don't get vaccinated, right? 99.9% death rate if you're not vaccinated after a rabies bite or a bite by a rabid animal. But, they won't tell you that, just like, RFK Jr. is vaccinated and so is his family. What's that all about? What are you going to do when you ban all vaccines, right? Oh my gosh. Alexis loves hantaviruses. We did a little short thing on Hanta when Gene Hackman died a couple of months ago in Santa Fe, right? Yep. Yuxing is in Hangzhou, China. 23C during the plum rain season at the turn of spring to summer. Welcome. Welcome. Mark Lamont on the east end of Long Island. Jeff is in Elgin, Illinois, where it's 89. Pretty warm. Is Amy still at NIH? Amy was not at NIH ever. She's at the FDA, which is an independent agency of the NIH. EHS Talent is in eastern Pennsylvania, where it's 25C. Just peace, folks. Which way do you do it? Like this? Richard Nixon. Nevitz, forever got his or her COVID booster. Ain't going to wait for RFK Jr.'s imbecility to figure something out. Yeah. Indeed. Mark Lamont is in East End, Long Island, 70F21C, uncomfortably elevated ozone levels. And Living River is now going to read the Living River. Just remember Living River. It's wrong, okay? But it's interesting to read because it's sometimes good to see how people are portraying things incorrectly. I'm guys finishing my virology lectures to prepare for my PhD defense. Thanks a lot for the videos. It's my pleasure to teach the world virology. Wish I could reach more people. We have this language barrier, but someday it'll be surmounted by instant translation. And so I'm glad we could help you. I really, really am very happy about that. This individual whose name I cannot pronounce since I don't know the characters. I've been watching Twiv from #1 recently. It seems like a nice way to remember Dixon. Back then you thought reaching 100 would be great, but unlikely. I didn't know if anyone would listen. Dixon and I started on September 2008. It was an experiment. That's what we do as scientists. We do experiments. I had no idea if it would work. I just wanted to try it. But people started listening. They started sending questions in, and the numbers just kept going up. So we kept going. And now we're at 1,200 something, right? Amazing. Amazing. Of course, I would not be here right now probably if it weren't for COVID, because we started doing live streams. Amy and I did live streams during COVID, and then Q&A with A and V became office hours, and so here I am. And many of you came for the pandemic and stayed for the chat, basically. Can you create a shadow CDC? So there is some talk about having the states have their vaccine advisory committees, okay? But here's the thing. The vaccine advisory committee is historically at the CDC, who does a lot of the research to test vaccines, right? And so they are definitive. They are the authoritative word, and therefore that's perfect. And so this current administration wants it distributed to the states. They don't like big government. You know what? I don't care what they like. It works this way, and we're going to get it back. Don't stop fighting, folks. Does ASV ever need on-site volunteers? I'm not a virologist, but I'm helpful and friendly. Well, they do have volunteers. Usually there's a hosting institution, a hosting university. So this year it's McGill University, and they get grad students and postdocs to volunteer. But I don't see why they wouldn't take a friendly and helpful Jill from Wilmington, Delaware. I can give you a contact for future meetings if you'd like. Roach, I'm in awe of what you do. Thank you. It's what I love to do, but I appreciate that sentiment. Peter is in Boulder, where it is 79.26 and roasting. Alexis, I've also watched my lectures to prepare for some upcoming stuff. I'm really glad to hear that. Could AI from Riley find a cure to prions? It could perhaps suggest some approaches, because everything that's been tried so far is not working. And those have to be drugs, right? Because it's a self-protein, so antibody is not going to work. So I don't think AI is going to solve problems like this. It can do other things, but they're just unknowns here that AI can't tackle, right? However, there is AI co-scientist put up by Google, which we covered on Twin a few months ago, and their specialty is to suggest experiments. So maybe it would work. Yeah. Carol loved TWIV 1225, especially the second article where we talked about, Omicron and immunity, antibodies and T cells. How about for measles, antibodies versus T cell immunity? That's a good question. I don't know. I'm going to look it up, though, and get back, and maybe next time we'll talk about it. Measles, mice, antibody, you could do knockouts. Yeah, I bet it's been done. I just don't know the answer. Very neat. By the way, there have been deaths from rabies because people refuse getting their vaccines. Of course. And there are deaths from people with HIV/AIDS. They don't want to take antivirals. They say HIV doesn't cause AIDS, and they die. Okay, here we go. The definitive thing tonight. 2 fingers with the palm facing out is a peace sign. 2 fingers with, I don't know what that's bad, I guess. 2 fingers. If it's reversed, it's a vulgar British insult. Sorry. Okay, this is how I usually do it. Yeah. Is it like FU or something like that? Thank you, Legotic. This is very important information. Peace and love. Peace and love, folks, all over. I can't do this. All over the world. Yep. Okay, what else we have here? Mark Martin. The river was important in my family because my in-laws knew Hillary Kaprowski's son, Claude. So I was to explain so much about minus 80s and PCR and immunology. There is a story. Let me tell you this story. I'll take Mark off here. So the idea was in the river that Hillary Kaprowski was doing polio vaccine trials in the Congo. And what he did was take chimpanzees, take out their kidneys, make cell cultures with them, and grow poliovirus in them. That's how vaccine strains were made. And they're made that way to this day, at least the OPVs. Although I think production has changed, actually, so it may not be non-human primates anymore. But this is the way they did it. And Hooper said what happened was the chimps were contaminated with SIV, the ancestor of HIV, simian immunodeficiency virus, and that got into people, and it started the HIV epidemic. It's not right. A lot of things don't jive. But what they did, they had some of these vials of the vaccine at the Wistar Institute in Philadelphia that were produced in the Congo and brought back. So I was tasked with aliquoting, dividing those vaccines into aliquots and numbering them so they would be blinded, right? And then they would do PCR to see if there were non-human primate, chimpanzee mitochondrial DNA in them, okay? So I met halfway, the director of the Wistar, halfway between New York and Philadelphia on the New Jersey Turnpike in the parking lot. We exchanged this white foam container with dry ice and vials of poliovirus. He said, this feels like a drug exchange here. And then he brought it back. They sent it to someone else to do the PCR, and there was no chimpanzee DNA in the samples. Okay, so that was the end of that. Pelican Nurse is in the Central Coast, and it's 23C. Welcome, Pelican Nurse. Yes, if you don't know how to pronounce Racaniello, it rhymes with Black and Yellow, the rap song, right? Black and yellow, Racaniello. It's very easy, very easy. Peace. Role of PCDH1 in hantavirus entry. What's the question? So it is a cell surface protein. Let's see. Hantavirus. It's the receptor for sure. It's the receptor for hantaviruses here. Receptor for hantaviruses. I think we covered that on TWIV, I'm pretty sure. So here's the title of the river, A Journey to the Source of HIV and AIDS. He says HIV infection in humans originated inadvertently from the large-scale evaluation of live attenuated polio vaccine in Africa. This is not correct. It originated from a spillover from chimpanzees to humans in Cameroon, and that was definitively shown by Beatrice Hahn. So this is wrong. Luther asks, how does the direct penetration of the virus into the cell occur? Well, it depends on what the virus looks like. So a non-envelope virus with no membrane like poliovirus. Here's poliovirus. This will bind to receptors, which is right here. This is, by the way, a Christmas ornament. It binds to receptors and is taken up by endocytosis, where the membrane wraps around it and pulls it into the cell. If you have a membrane, the membrane can either fuse with the plasma membrane of the cell or be taken up by endocytosis and then fuse later on in the endosome. Those are the different mechanisms. Lecture number 5, my virology course. Mario, my 9 and 11-year-old boys are watching you. They say hello. Hello, 9 and 11-year-old boys of Mario Vera. Very nice to see you. David says, use the SIBDRAP site for medical news, including some virus content. University of Minnesota. Yes, so they are working on gathering those pushed out at federal level. They've actually established a vaccine advisory committee of their own. And so I don't know. They got funding for that, and I don't know how it's going to work. I think it's just still being worked out, but we talked about that a few weeks ago on TWIV. Hello, Will. Good morning from the hospital in Changsha, 24C. Hope you're okay going home this morning. Hope everything is okay. Glad to have you here. Jeff says, no, AI will not find a cure for prions. There's no training data for that. However, they may be trained on proteins to help identify target regions. Yes. Yes. Lurie is in Pueblo, Colorado, where it is 31C. Aubrey is in Richmond, Virginia, where it is 26C. Yes, the struggle indeed continues. Mario, what's the latest on that chick vaccine? Well, it was paused. Its use was paused in people over 65 by the FDA, pending evaluation of reports of serious adverse effects in that age group. So it's going to be a while before those data are sorted out. Truth or dare, 55,000 deaths per year from rabies in India and Africa. Yes, it's huge. Huge in other countries. And it's vaccine preventable, right? Javier is in southern Ontario where it's 20C. No reason to apologize ever. So this can be peace or victory, but I am using it as peace. Peace, folks. I mean, really, I wish we could have peace. Ay, ay, ay. Mark says, you don't tick me off. I don't know why you're saying that. Did I say you did? I never said you ticked me off or I ticked you off. No. What's that from, Mark? What's the context? The river monkey kidneys, not chimps. No, it was chimp kidneys. That's what they were using back then. They don't use them anymore, obviously. It's just Kaprowski used chimps at the time. You could do that. It was a long time ago, right? Mateo, how was your week? Personal advice, I applied for some PhD in virology, has gotten none. I'm beginning a master's. What advice do you have to get into a PhD? Well, if you're doing a master's, that's great. Do it in some science that's relatable to a PhD, like biochemistry or cell biology, right? Something like that, even virology. And if you got some research experience, that would be great because admission committees are always looking for that, right? Not just writing a thesis, but actually doing some research. Hello, Zhu. Welcome back to 81C in Austin, and thank you for your contribution to science communication. You know, here at Microbe TV, we are a nonprofit, so all your donations are U.S. federal tax deductible, and we don't do advertising, so we depend on you for our production costs. So if you like these programs, please donate. Yes, and Michael Warby and has written extensively on HIV not coming from poliovirus trials. Yes. Viruses, so incredibly simple yet so smart. Well, I don't know about smart, right? But they are simple and they do amazing things. Do you have Usutu over there in the U.S.? No, we do not. Related to West Nile, a flavivirus. It's killing off our beautiful blackbirds here in the UK. It's been spreading via mosquito bites. That's too bad. Let's see. Let's just make sure. Nope, hasn't been found in the U.S. yet. Okay. Krishnan from Malaysia, good morning. Happy to have you. A little clarification, the peace sign is actually the victory sign from World War II, and the peace sign symbol is the logo of the Campaign for Nuclear Disarmament. But I guess this has been usurped for peace, because I'm not declaring any victory. I don't need to beat anyone. I just peace, folks. Love each other. Don't be nasty. Be nice. Okay, Mark, goodbye. Have a good dinner. See you later. Here's the virus entry lecture for the person who asked about entry into cells. Thank you, Les. Interesting. We have blackbirds here every year, but I've noticed none for the past couple of months. I really miss their song. That's really too bad. Okay, next question. How do rabies vaccines work? From the Living River. So there's an inactivated or attenuated rabies vaccine, right? The inactivated, they're both injected, and the inactivated does not reproduce. The attenuated does reproduce, and then they both induce antibodies and T cells, which neutralize the virus and prevent it from invading the central nervous system. And the thing about rabies is that it takes so long for the virus to get from the bite site into the central nervous system that you have time to vaccinate. You have a couple of weeks, depending on where the bite is. Now, if the bite is on your face, it's tough because the face is close to the brain, if you have one. Many people in this country seem not to these days. But I digress. If the bite is on the face, you don't have a lot of time. If it's on your leg, that's great. So the next time you have an encounter with a rabid raccoon, have it. If it's got to bite you, have it bite your leg, not your face. Don't get it in its face, okay? With respect to prions, antibodies can't be raised against the misfolded protein. Aren't they conformationally distinct? They are. They're very conformationally distinct from the normal protein. However, there is no immune response against it. So it's not sufficient, although people are trying to figure out how they might do that, right? But so far, no antibodies against the misfolded protein. Questel is late because she was jamming at the stage at the club. So in the club, you were a star, and now you come and you say you're hiding in the corner. No, you're not hiding in the corner. You've been here since day one. So what do you play? Tell us about it. And wow, Questella is a rock star. Wow. Questella dances. That's cool. Got some good stretching in. Good for you. Stretching is good. Yeah, Blackbird. Beatles song. Yep. Bye-bye, blackbirds. Yep. How is the cap stolen from cellular RNAs? Sorry. So the virus encodes an enzyme that cuts the mRNAs. It's part of the polymerase complex. It cuts the mRNAs, a certain distance from the cap, and then those RNAs are used by the polymerase as a primer. How does rabies transport from the bite site to the nervous system? By neural pathways. So it gets into your peripheral nerves at the bite site and then goes up, because remember, all your muscles are innervated and they go all the way back to the spinal cord and eventually to the brain. There's the primary neuron in the brain that innervates that muscle. So the virus goes by those pathways. Etoy 2001, hello from Cincinnati. Good to see you. Mario, do you think national measles cases and deaths will go any higher? Case numbers are approaching those of 2019, and it's only the middle of the year. Okay, so here's the story. More and more parents are choosing not to vaccinate because they have encouragement from RFK Jr. They say, oh, look, the head of HHS tells me I don't have to, it's my choice whether I vaccinate my kids or not. So many parents are choosing not to. So they're going to be more and more susceptible. So I think we'll have a lot of cases. Now, according to Paul Offit, natural measles usually stops when there were outbreaks in the U.S. They stop in May, but this is not stopping. I don't know why. Nevertheless, there are going to be a lot of cases, and then when the season begins again in the fall, there are going to be more. We're going to have huge numbers because many, many people are choosing not to vaccinate their kids, and many more will not, okay, because of this guy saying don't vaccinate. Or he has said not to vaccinate, but he also will say, it's your decision. You decide. Okay, so Churchill, more on the peace sign. So we started with a peace sign, and now we're getting a lot of discussion. Churchill adopted the peace sign in the middle of World War II. V became a symbol of resistance in occupied countries. Very cool. Welcome, Miss Blue Ocean. Polka dot puck. Down in Texas and Oklahoma, there are plenty of anti-vaxxers. However, everybody gets their dogs vaccinated for rabies. I wonder if they have ever thought about that. Yeah, what about the voice of the dog, Everyone says, I want a voice in whether I get vaccinated or not. What about your frickin' dog? Huh? Maybe your dog doesn't want to be vaccinated. They don't think about that. They think it's an animal and it has no sentience, right? That's not true. Dogs are cool animals and they are aware. And when you stick a needle in them, it can hurt. So they haven't thought about it. Do you think anti-vaxxers have thought about anything except my right to put in my body things, foreign substances? No, it's totally inconsistent. I didn't know this. Beethoven's fifth opening note is V in Morse code, so the BBC played that too. Interesting. How interesting. So in a couple of years ago, I got an award in Switzerland, right, in Zurich, and at the award ceremony, they had a 4 or 5 member French horn combo. It was a quartet or a quintet. And they wrote a composition for me called CD155. That's the name of the poliovirus receptor, which was identified in my lab. So the first 5 notes are CD155. whatever 155 is. Isn't that cool? I have the music. It's great. I love it. Oh, this is good. Having some states not vaccinated is like having a peeing section in a swimming pool. That's great. I love it. I love it. Dogma, should I get my 85-year-old father with dementia vaccinated again as COVID is back in town? Well, that's the CDC recommendation, right? Anyone over 65 can get vaccinated with the current variant booster. And so Daniel Griffin agrees that that's a good practice. So yes, you probably should do that. Not giving medical advice. I am simply repeating what the CDC and Daniel Griffin has said. I think measles now is worse than the 50s and 60s. Now, there were many more cases in the 50s and 60s. Pretty much all the U.S. population was infected. But, you know, aggressive vaccination campaigns, including school mandates, eliminated the disease by 2000. And now it's come back. Yeah, dogs are people too. Miss Amy. Never thought, has there been attempts to design rabies to reach nerve cells for positive medical reasons? Would like to hear a twiv about something like this. This is actually a good idea. I like that. Certainly, so rabies is used as a neural tracer for tracing nerves because it travels within nerves. So you can put a dye in it and watch it travel and map nerve circuits. And I'm pretty sure it's being explored as a therapeutic vector. Yeah. Visto's puppy is not anti-vax. He's smarter than most in Congress. Yes. Kang says, choosing not to vaccinate is not an informed decision. No, it is not. In fact, they say it's informed, but what they're doing is listening to the anti-vaxxers to give them their information. They don't listen to us, the pro-vax people. So figure it out. Microbe TV. Could the rate of measles infections be due to too many children not playing outside as opposed to being indoors? No, I don't think so. I think the majority of infections are in unvaccinated children. You don't need to posit any, hygiene hypothesis stuff to explain that. Okay, Peter says CD155 and TVC15 transmission. I don't understand what TVC15 is. I'll look it up, right? TVC15. Oh, it's a David Bowie song. Lyrics. Okay, it's a David Bowie song, but I don't know the connection. Can explain it. It wasn't an orchestra. It was just 4 or 5 French horns. And they were played by people who were scientists, these French horn players, right? So it was good. I loved it. It was a great honor. So I have to tell you, this is the Richard Ernst medal that I got a couple of years ago for my work in science communication. Richard Ernst was one of the 3 developers of the MRI. Can I do 3 like this, right? It's probably best to do this than this. 3 developers of the MRI. And he realized that, doing something like that, you have to communicate to the public about it, what the impact is, it is, and so forth. So he started this lecture series about people who communicate their work to the public. And previous winners included Kofi Annan and Stephen Chu, right, the physicist, and me. And I don't know why I got this, but it was for my podcast, obviously. So I went over there and gave a lecture and got this very nice medal. And the CD155. Oh, excuse me. It was very cool. Very cool. Thought Seer. Livestock are 100% vaccinated. They are also at the center of all pathogenic outbreaks. Example, when and where mass slaughter takes place is when and where timing of flu outbreaks. There's more. Okay. Marcela Vega, I'm truly grateful for your contribution. I listen to each of your channels. I check the articles. I'm convinced your voices will resonate. That's the kind of confidence that we need to have. Yes, that our voices will resonate. Roach says, by measles, worse. I meant more virulent now. I don't think it's any more virulent, no. I think same virulence, yeah, looking at the numbers, of like pneumonia and deaths and so forth. Worm Talks 94, thanks for giving me a shout out to my YouTube channel on your last TWIP. Of course, we like to publicize good science everywhere. Everywhere. Also, I'd like to see another episode of TWIV 496. It's such an amazing resource, but it's 7 years old now. TWIV 496. TWIV 496. Let's see. Isn't it cool? I can type TWIV 496 in and Google finds it. Vaccines work whether or not you believe in them. Yeah, that's actually a very good episode. I wish people would watch it. It was very compelling. But, all the anti-vaxxers won't watch it. They won't believe it. We never achieved herd immunity with natural measles infections in the 50s and 60s. Yeah, it continued to spread mainly because children were born all the time, right? And that gives new hosts for infection. As soon as you had enough kids, you would have infection. But you're right, the vaccination rate was not as high as it became after school vaccine mandates were put into place, right? That's what really did it. Yeah. Living River says, I deserve the recognition. Your podcast has given so many people a passion for science. Well, I was very humble about these sorts of things, right? Because I did this as an experiment, and it turns out to work to teach people things. So I appreciate the recognition. It's great. Peter says the TVC connection was the only phonetic similarity in the titles when saying the individual numbers. Okay. Thank you. All right, here's some news items, okay? First of all, this is a CNN article, so I should be able to show you this. So let's share the screen. RFK Jr. removes all current members of the CDC Vaccine Advisory Committee. All right. He just fired them all, 17 members with lots of experience. So these individuals advised CDC on the vaccine schedule and required coverage for different people. And he said he has the authority to do this, but usually you don't take everybody off at once. So the panel's members are pediatricians, epidemiologists, immunologists, physicians, and they're not supposed to be political appointees. But Kennedy claimed that the current committee was rife with conflicts of interest, but the conflicts were recently published in 2024, and they don't have any conflicts of interest. So where does he get this data? Show us the data. I got Pete up here. Let me turn him off. Sorry. Now we have an ad. Great. Here, I've never seen anything this damaging to public health in my lifetime, the advisor said. I'm shocked. So this is someone who would not be identified. So Bill Cassidy here, the Republican senator from Louisiana, he's an MD. Kennedy had previously pledged to consult with the senator on filling key roles on vaccine advisory boards. But he didn't do that. And Cassidy says, of course, now the fears that the HCIP will be filled up with people who know nothing about vaccines except suspicion. Great. Well, look at Susan Collins, who voted for him. Seems to me excessive to ask for everybody's resignation. Really? Is that the only thing you can think that's wrong with it? All right, so now he's appointed some people, and I can't get that article up here because it's a New York Times. Let's see. Where is it? Here, 8 new members, okay? He's named 8 new members of 8 doctors and researchers, including 2 prominent critics of federal... Let me take this off. I don't need this anymore. Of federal scientists in the Biden administration's COVID vaccine policies to replace roughly half the members he fired. He says the slate includes highly credentialed scientists, leading public health experts, and some of America's most accomplished physicians. The 2 of them, Martin Kulldorff and Robert Malone, are likely to draw an uproar from both pro-vaccine groups. Both were highly critical of Biden's corona vaccine policies. Kulldorff is a Swedish biostatistician. Former Harvard professor who used to advise the CDC. He was one of the co-authors of the Great Barrington Declaration, which said, "Let it rip." Great. Dr. Malone played a role in early mRNA research and is pissed off that he didn't get a Nobel Prize. Then in 2021, he appeared on the Joe Rogan podcast and spread all kinds of misinformation. It's just what we need. Those are the only 2 I know. I don't know anyone else who is on this committee. All right, so that was one thing. And the other is just very disturbing. I think I can show this to you. It's a Science article. NIH terminates network aimed at stopping pandemics before they start, because they think it's unsafe to identify viral threats. What kind of world are we in now where people are so stupid? It's unsafe to identify threats. So what? You just stick your head in the sand and you don't do anything? It's ridiculous. So there were a network of centers for research in emerging infectious diseases. 5 years ago, they had an $80 million budget to characterize and collect mosquito-borne viruses and pathogens that could jump from animals to people. But they were told to stop working. They said, current agency priorities do not support this work. Assholes. I'm sorry. That's just a BS excuse. Current agency priorities. You don't have any priorities. That's the problem. So here you are collecting viruses to see which are potential pandemic pathogens, and you say you can't do this. It's not safe. That is just dumb. And it's just politically motivated by all these people who are saying that virology research isn't safe. And it drives me nuts because nobody asked me. I did virology research for 44 years, and I can tell you it's safe. You take proper precautions. So the people who could criticize it have no idea how this works. So anyway, David Wang, who's at WashU and led one center, calls the claim that this work was not safe totally unsubstantiated. In fact, if we can detect and stop a virus where it starts, that directly makes America and America's citizens safer. The cancellation is incredibly short-sighted. By canceling the network, NIH is pandering to the lab leak conspiracy theorists. That's absolutely true. So, folks, those of you listening, all 200 of you, by the way, hit the like button, please. We are in big trouble in this country. So the Vaccine Advisory Committee rejiggering eventually will lead to unavailability of most vaccines. And there'll be huge numbers of infectious diseases coming back. And this, we aren't able to predict pandemic threats. So we're totally vulnerable again, which is ridiculous. If you like that, okay, well, you're wrong to like it. But, write your senators, write your congresspeople and tell them that this is unacceptable. These cuts, which are made with no brains, just politics, right? Science by edict, not by committee. So anyway, let's see. We have some more comments here. Okay, I have to go back up. There's a lot of comments since I started. Okay, hello, Jonathan from Encinitas, 18 degrees C. And Aubrey says, I understand the outbreak is due to lack of vaccination, but you had stated the rate of infection slows during the warm seasons, according to your statement. It isn't. Could this be due to too many indoors? I don't know. I mean, you're saying that kids' immunity isn't good enough, right? But, I mean, the kids are not vaccinated, right? So, yeah, I don't know why it's not slowing. Maybe it will slow in the next weeks, and it's just a statistical blip. I don't know the answer, and I don't know if it's due to, exposures or not. Bad Cat says Bad Cat voted against eliminating school mandates. School mandates are a great way to prevent infectious diseases, right? And they worked in bringing measles to elimination. RFK Jr. appoints Mr. Fox as head of the chicken coop protection team. So former ACIP has joined Sanofi. Yep, they need to have a job, right? Feels like the U.S. is circling the drain. Yes. Malone is a charlatan and RFK Jr. is a liar. And the lunatics are running the asylum. Ms. Blue Ocean, Sandy, sorry. This bunch of morons are also taking the climate.gov site down. That is one of my favorite go-tos. Pete says, sadly, you also got one of our UK nutters, not on the vaccine committees, but an advisor elsewhere. Carol's daughter, going into pediatrics, states that the majority of pediatricians will follow vaccine guidelines from the American Academy of Pediatrics. Yes, that would be good. RFK Jr. makes me think that if the phrase every accusation is an admission with regard to conflicts of interest. So RFK Jr. is incredibly conflicted, okay? One sec. He's working with a law firm that sues vaccine manufacturers. He advises them, and he takes millions of dollars a year from them. That is a conflict of interest if you're heading HHS. If you're too dumb to recognize that, then get out. And he's saying that the ACIP members have a conflict, and there are none. He's a bloody liar. Don't you get it, folks? Not you, but everyone out there who voted for this friggin' administration. We're in the looking glass world where the morons rule. That unsafe to identify viral infectious idea is typical for the Republican Party now. GOP populism will destroy our liberal society if rational people don't get active. RFK Jr. has named some pandemic nitwits, Malone, Kuldorf, and Bhattacharya, the NIH head. Senator Ernst is right. We are going to die sooner. Someone said that USA citizens could face being banned from entering countries as this progresses. No! Don't do that. I want to travel and do podcasts now. Oh, well. The 496 topic is vaccines work whether or not you believe in them. Thank you. Thank you very much. Senator Ernst left out the part where they're going to be the cause of our deaths. Yeah. This whole Congress, they have no spine. They knew how bad RFK Jr. was. And they voted him in. All the Republicans could not say no. They're just chicken. Unbelievable. Will says, back in 2017, Xi Shi Zhongli at the Wuhan Institute of Virology warned that bat viruses could result in a pandemic, and they have, and then she gets blamed for it, which is ridiculous, right? RFK is at your cervix, should you have one. The HPV vaccines prevents cervical cancer as well as oral, anogenital. RFK Jr. gets $2 million if he wins his suit, and women get cancer. I don't know how the HPV vaccines are completely safe. He says it's the most dangerous vaccine around. It's just a lie. He's making money off it, and he doesn't care if women get cancer. That's this administration. No humanity at all. They don't care about anyone but themselves. Dogma says it isn't just the U.S. that is circling the drain. A lot of research decisions affect lives in India, and we have some anti-vaxxers here, too, who will pick up any idiotic stuff from the U.S. that they can quote. Yep. Totally. One more, and then we'll do a mini lecture. CO2 breached 430 parts per million this month. Pre-industrial was 280. Yeah. But, of course, it's not a problem, according to the other party. Okay, let's see. Mini lecture. Evolution Part 2. Good, looks good. Let's hit it. All right, remember last time we talked about all the mutations that accumulate when viruses reproduce. That's a big part of evolution. But things will not evolve unless you have selection. You have to select out the mutations that give the virus an advantage. And so we're going to talk about two examples of selection: genetic shift and genetic drift. And so when a virus is reproducing in the face of antibodies or cytotoxic T cells, those mutants that are resistant to the antibodies or T cells will be selected. And that's what we're going to talk about here. There are two names for this. There's drift. This is diversity that arises from the polymerase making mistakes, and then the antibodies or the T cells selecting for the variants that can overcome the antibodies or the T cells. So the viruses are resistant to the antibodies, for example. This is the case with many of the SARS-CoV-2 variants. Or shift. This is diversity arising after recombination or reassortment. So it doesn't involve mutations as drift does. So let's contextualize this, as they say. We'll use influenza viruses as an example. So as you know, these viruses are classified by serologic testing of the two viral glycoproteins on the surface, the HA and the NA. So we call H1N1, H2N3, etc. So there are 18 hemagglutinins and 11 neuraminases. All of these hemagglutinin-containing viruses from 1 to 17 can infect birds, and H1, 2, and 3 can infect and transmit between humans. Okay? Only the H1, H2, and H3 bearing influenza viruses will become human viruses. H5 infects humans. It doesn't transmit very well at all among humans. If it did, then we would add it to this list. Okay? That's the background we need. So the HA protein is shown on the right. And this is going to illustrate what we call antigenic drift. Those colored areas are places where antibodies bind to the hemagglutinin. So the hemagglutinin is on the virus particle. It's the blue thingies here on the left, the blue spikes. Those changes in those colored regions can interfere with antibody binding. And so if you have an immune response and the virus changes, it won't be neutralized well by the antibodies. And so that's an example of drift. Let's say a virus just randomly arises with an amino acid change at one of those sites. Now antibodies don't neutralize it well, and so that overgrows all the other viruses which can be neutralized. So that's an example of selection. The antibodies are selecting for viruses that are not neutralized by the antibody. They're allowing them to overcome the population. And one amino acid change at some of these sites is sufficient to make us have to change the vaccine from year to year. Okay. Same thing with SARS-CoV-2. It has a spike protein. Here's the spike protein structure on the right, and it's shown with the red molecule is ACE2, the receptor. So this interface between the spike and the receptor is grounds for change. There have been a number of amino acid changes at this site, and these are just 3 of them, which block antibodies from neutralizing infection. So the neutralizing antibodies tend to act at the receptor spike interface, and they block attachment, so they block infection. And antigenic drift occurs at these sites. The antibodies cannot block anymore, and the viruses can reproduce. And we've been tracking variant SARS-CoV-2 for a long time now, right? And here's an example of sequences of the preceding 2 weeks from different countries. And you can see the percent of the different variants. So here are all the variants at the top, right? You got your Delta and Alpha. And here's an example of how in many countries Delta is predominating. This was 2021. But you see Omicron in red is creeping up. And in fact, in South Africa, where it seems to have originated, it's now all of the isolates. All of the sequences in this 2-week period are Omicron. So these viruses slowly displace previous ones. Now, a little bit of a diversion here. Why do they displace them? Well, people say they're more transmissible, but is that really true? Here's a comparison of SARS-CoV-2 with influenza virus. What do I have here? These are the percent of different variants at different times from 2020 to 2021. The variants are given different colors. Right, so there's a different naming scheme here, but you can see the early 19A variants displaced by 20A and B, and then this variant here on the right arising and displacing them with time. The same exact thing occurs with influenza viruses. These are 3 different plots of similar data for H3N2, H1N1, and a B isolate. You can see displacement of variants. There's no evidence for increased intrinsic transmissibility for these influenza viruses. They are all immune evasive, and it's same with SARS-CoV-2. There's no evidence for increased intrinsic transmissibility, but that's what people say immediately when they see these variants, okay? So that's antigenic drift. Now, antigenic shift is a different animal. Influenza viruses have 8 RNA segments as their genetic material, and when those infect a cell, they can reassort, they can mix. If you have 2 different viruses infecting a cell, they can swap segments, and out comes what we call a reassortant, basically a new virus with new properties potentially. And so this is a diagram of recent influenza virus pandemics and the viruses that caused them. So remember, 1918, we have a big pandemic killing millions of people globally, bigger than SARS-CoV-2 pandemic. And this virus that caused this was thought to have come directly from a duck influenza virus into people. And so this duck influenza virus, I'm showing all the segments of influenza virus in red. So this 1918 pandemic virus had all of these avian influenza virus genes. That virus circulated until 1957, and then it was displaced by an H2N2 strain, which caused another pandemic. And this one got 3 new segments from a different duck influenza virus by reassortment. And you can see those in yellow here. The HA, the NA, hence the change from H1N1 to H2N2, and a PB1, which is part of the RNA polymerase complex. So nobody had immunity to these duck influenza virus HA and NA gene proteins, so pandemic. That virus circulated until 1968. It was replaced by an H3N2. Again, we got some influenza virus segments from a different duck influenza virus, and you can see those in blue here. But the HA changed, and again, the population did not have immunity to H3. pandemic results. And again, all these viruses, 57 and 68, arise by reassortment of two viruses going into a cell and swapping genes. Viral sex, unprotected sex. And then we have our 2009 influenza pandemic, the H1N1, and that's the last influenza pandemic. So we're due for one, right? If you look at the year intervals here. Here's the virus here on the right. Look at all the different colors of the segments. They came from so many different ancestors, starting in about 1990. This pandemic was 2009. This virus, this influenza virus that caused the human pandemic, derived RNA segments from influenza viruses that infect Eurasian swine, classic swine, human H3N2s, avian influenza viruses, and North America swine. This has all been mapped out very nicely. And so that's why we have so many different RNA segments making this up. So this is called antigenic shift, because all of a sudden, boom, you have an H2N2, which has not been around for as long as we know. Nobody has immunity to it, and you get a pandemic. So currently, this H3N2 and the 2009 H1N1 are co-circulating. H2N2 has not circulated since 1968. So anyone born after that has no immunity. There are lots of people born after that. And so if that virus is somewhere in some duck in the world, it could be reintroduced and cause a pandemic. I think that's a good scenario for the next influenza virus pandemic, an H2N2 strain. Okay, that's antigenic drift. Next topic, Red Queen conflicts. What's that? Lewis Carroll, Alice in Wonderland. The Queen says to Alice, around here it takes all the running you can do just to stay in the same place. What does that mean? Here it is. We have a virus in green and a host protein that can bind the virus and block infection. You're inevitably going to have selection of virus mutants that evade the host protein. I just showed you an example of that in drift. But the host can change too, even though it's slow, and it can readapt and block infection with that virus. The virus can go, the cell protein, this goes back and forth over geologic time. And that's why it's called a Red Queen conflict, because you just go back and forth. It takes all the running you can do just to stay in the same place. Whoa, it's great. Good job, Lewis Carroll. We can study this. Beautiful way of studying it. So let's take primates here. And we have a phylogeny of different primate species, right? A tree. And we have a gene that we think is a cell gene involved in virus defense. And we have the sequences of that gene. So these are orthologs. Those are the proteins that are present in the different primate species. We then use a computer program to identify positively selected lineages and codons. What does that mean? Positively selected. We analyze the ratio of non-synonymous to synonymous changes. Remember, the genetic code is redundant. Some amino acids are coded for by as many as 6 codon triplets, right? So you can have lots of change occurring over time, which is synonymous. It codes for the same amino acid, but the non-synonymous changes code for a different amino acid. And so you can analyze the ratio and identify codons that are positively selected. And you can see here, there seems to be some amino acids that are positively selected in specific lineages of this phylogeny. So in other words, those are candidates for changes that are responding to a virus. And then you can check it. You can look at cells in culture from the different species and infect them and use the right sequence, and you can see if the host wins or the virus wins, depending on the sequence of the virus and the host. And then you can map these changes on the virus host interaction. You could say, for example, that when the virus wins, it's a certain sequence, but then when it changes from S to G, then the host wins. And you can notice that in the sequences of these proteins over the phylogeny. And so by doing this, we have been able to map these virus-host conflicts and how, for example, they've driven the evolution of the immune system. So here are two proteins in the immune system that are involved in virus defense. We have TRIM5 alpha on the left, which antagonizes the HIV capsid. And you can see there are positively selected amino acids here in this domain that go back and forth with virus changes. This protein on the right is very interesting. It's PKR. It has domains that have been selected for amino acid changes in different domains that have been selected for by various viruses because it's a very broadly acting antiviral protein. And so you would look at this protein having several surfaces that bind different viruses, and those can change as well as the counterpart on the virus. So it's really amazing that you can use these sequences, the phylogeny, to map these Red Queen conflicts, essentially. Another example would be the transferrin receptor, which is cell surface receptor that binds transferrin, which in turn binds iron, which is essential, right? It takes it up into the cell. And two viruses have driven evolution of the transferrin receptor, an arenavirus, and those residues on the transferrin receptor that are involved in defense against arenaviruses are shown in gray, and then mouse mammary tumor virus, which are shown in blue. Now, this is the receptor for these viruses. So the receptor has changed over time to prevent infection, and then subsequently the virus has changed as well. And we can map those residues here on the protein that are involved in binding to the virus. Now, interestingly, the transferrin binding region is here, so it's not affected by these changes. If these changes to prevent virus binding prevented transferrin binding, they would be lethal, right? You couldn't live. So the changes, I don't know how this happened, but the virus binding site is at a different place from where transferrin binds. If it weren't, you wouldn't be able to change transferrin to avoid infection. Okay, so I've told you today and last time, we have enormous genome diversity, right? All these mutants, but there are 3 serotypes of polio. There are a bunch of rhinoviruses, but there's 1 serotype of measles. Yeah, a lot of influenza virus variation. So what's with polio and measles? Well, in the lab, we can show that they can undergo antigenic variation, but they rarely do so in humans. And it's most likely because there are constraints on the viral proteins that limit the amino acid changes that give you a viable infectious virus. Okay, another kind of selection I want to talk about is virulence. Is virulence a positive or negative selection? What do I mean? Positive trait means you're selecting for higher virulence. And this could be because high fitness is selected for in virus revolution. What is evolution? What is high fitness? Just the ability to make more virus. And so a positively selected trait of fitness could result in virulence because you have more virus. Virulence could be a negative trait. Maybe if you have a more virulent virus, it kills the host too quickly and it inhibits transmission. Or it could be a neutral trait where it doesn't do anything. Maybe there's some other phenotype that is selected and that gives you the effect on virulence. And so I think most likely all of these are in play. Very hard to answer it because we don't have any real world examples. Like HIV spilled over into people in 1920 and it's still quite virulent. So we haven't seen any changes in that and we can't correlate it with sequence because there are no changes. But there is an experiment that we have done in the evolution of virulence, which gives some insight onto whether virulence is a selected property or not. So this begins in 1859, when the European rabbit was introduced to Australia for hunting purposes. So the rabbits that live in Australia were too quick for the hunters, so they brought in these slower European rabbits for hunting, and within years, because there are no predators, they reproduced to plague proportions, as you can see here. They had a problem, a population problem with rabbits. So what did they decide to do down under? They released a virus, myxomavirus, in the 1950s to rid the continent of the rabbits. So the natural host of myxomavirus is the cottontail rabbit. The virus is spread by mosquitoes. The rabbits get warts. They're fine. They don't die. But European rabbits, which are the ones released in Australia, are a different species, and infection with myxoma is 90 to 99% fatal. So they said, let's kill the rabbits with myxomavirus. So they released the virus. In the first year, the released virus killed 99.8% of the rabbits. But those 0.2% of the rabbits, did they throw a wrench into the works. In the second year, the mortality dropped to 30%. So the rate of killing was lower than the reproductive rate of the rabbits. Couldn't eradicate the rabbits. Rabbits reproduce a lot and quickly, kind of like viruses, not as much. So they couldn't eradicate it. What happened? Both the rabbits and viruses, of course, produce large numbers of offspring, so evolution happens quickly. Variants of the virus were selected that kill fewer rabbits and extend the life of the lethally infected rabbits so that the virus can go over the winter and spread in the spring by mosquitoes. And also the rabbits evolved to become more resistant or tolerant of the virus. And this has been confirmed by sequencing of the virus and the rabbits. And this is almost what you would predict for an evolving host coming into equilibrium with a new pathogen that spills over into it. We introduced the virus into Australia. It had never been there before. It had never infected these European rabbits. And so it comes to some kind of equilibrium. In this case, balanced virulence. Lower, but not zero. And what about in people? Can we see evolution of virulence? Well, it's very hard, as I said. So we have had a number of viruses spill over from animals into humans, right? Lassa virus, Ebola virus, HIV-1. They're initially quite virulent, and they're still quite virulent. But viruses from older jumps, like measles and polio, are not as virulent as those others. They kill fewer people. Not to say that they're not as problematic. But did something happen in the meantime, in all those thousands of years since measles and polio virus spilled over into humans? Don't know. We don't have the original viruses. There's no way of knowing. Now, SARS-CoV-2 is very new, and we really don't have any convincing data on changes in virulence. Many people make observational studies of hospitalizations and so forth. We did a paper on that on TWIV, but they're fraught with problems. And so virulence is very hard to measure in humans. You can measure in an animal model, but it's not clear what that has to do with people. So it's a real conundrum. But of course, population immunity has a big effect on virulence. And a great example are the common cold coronaviruses, which have been around for many, many hundreds of years. They originally spilled over from animals, caused pandemics of severe disease. We don't have records of them, but we assume that happened. And today they're still around, and they cause a mild upper respiratory tract disease. Why is that? It's not probably because the virus has changed, although we don't know, because we don't have the original virus. But you have a population of mixed older and younger people. The virus tends to wipe out the older people. Infection at a young age is less severe than an older age. So with time, you have no more older people who have never seen infection. The young people get older, they have immunity, and when the virus causes an outbreak, The older people are protected, and the younger people are as well. So this apparent evolution of virulence is really the evolution of population immunity. But we're still obsessed with increased virulence. It drives me nuts. The headlines: Ebola is mutating. Ebola, is it getting better as it's replicating from person to person? Peter Hotez says Zika mutated. No! It's easy to blame mutations, but there are usually other explanations. SARS-CoV-2 variants of concern are suggested to have increased or decreased virulence. In my view, unproven. You can't prove it, and these headlines are just wrong. We have no data on the effect of evolution of viral virulence in humans, but I can tell you transmission is a major selective force for viral revolution. How do you transmit? By making a lot of virus in the person who's shedding. So really, selection is in the end for fitness. Fitness is increased reproduction. If you're fit, you will transmit to another host. And whether that makes you more virulent or not, it could be. So these variants of SARS-CoV-2, in fact, have increased fitness. They make more virus. What makes them more fit? Often immune evasion for Omicron, right? They reproduce just like influenza virus in the presence of antibodies. But we don't know, other than that, what other kinds of properties are conferring increased fitness. That's why these variants of concern, or just variants, take over and displace older variants, because they have higher fitness. They reproduce to higher levels. All right, finally, let's take a look at the origin of viruses. Originally, we didn't have very old virus stocks. 1918 influenza virus, Pandora virus here, 50,000 years of age. But we developed techniques to estimate the age of viruses. So we have molecular clocks. And in this example, we can relate herpes virus genome evolution with that of the hosts, and we can determine that the three major groups of herpes viruses arose between 180 and 220 million years ago. That would put them right here, Triassic, Jurassic Park time. Dinosaurs had herpes, folks. Oh, we don't know that. They had herpes viruses. Can you imagine the size of a cold sore on a dinosaur? Another technique is by doing phylogenomics. So we have here 3 species of rabbits, okay? And we have a phylogenetic tree showing that they all arose from a common ancestral host over time. And we know that there was a split at some point, and these A and B derived from this branch on the left and C on the right. These rabbits, their genome can be sequenced, and we can look at endogenous viral elements, pieces of viruses that have integrated into the genome. Turns out that species A and B have the same Eve integrated in the same place. That means that at some point between the ancestral host and the split, that virus infected this lineage, leading to species A and B. Now, species C does not have that viral integration. That implies that the ancestral host did not, right? Because the ancestral host gave rise to all 3, and therefore the maximum age of that virus is here, and the minimum age is here. So you can get estimates for how old viruses are, and people have done this, for example, for single-stranded DNA viruses in different species. And we can tell that they were circulating, 30 and 60 million years ago by this kind of analysis that I've just told you. So how old are they? Well, some other analyses have, especially for retroviruses, estimate that they are over 450 million years old, and they originated in the oceans, infecting these cephalopods such as this. So 450 million years ago, Ordovician period, when the first land plants are arising. But, the ancestors of viruses likely arose before cells. We think in the pre-cellular world, so on the left here, there are no cells on Earth billions of years ago. But nucleic acids evolve, and these nucleic acids can replicate themselves. They can copy themselves without proteins. And so there's extensive evolution of these, what we call replicons. They are RNA and possibly DNA replicons. And, at some point, cells arise by a different evolutionary mechanism. And these replicons invade the cells. They steal capsid proteins and they become true viruses. And then as the cells evolve, they go from protocells to modern cells. The viruses evolve and diversify as well. So we think viruses originated from this pool of replicators many billions of years ago, before cells were even around. Not true viruses because they don't have capsids. So all the viruses that infect humans likely evolved long before humans appeared on Earth. So here we have Homo sapiens here in its brief stay on the planet. And all its ancestors. And so it's likely that many of these ancestors passed viruses down eventually to us. And so between this and the viruses we get from other animals, we can certainly say that all human viruses evolved from animal viruses. So how do you get a new virus? Currently, we assume that new viruses only arise from viruses that are in existence, not de novo. We don't have any brand new viruses evolving today that we've never seen before. So let's do a thought experiment and see why this is the case. What's the number of all possible mutations of a viral genome, and could it mutate to become a different virus? So let's just take an RNA virus genome. We've done experiments to show that over half of the nucleotides can accommodate mutations, okay? So for a 10,000 base viral genome, that means 4 to the 5,000 sequences are possible of that genome. So if you can mutate half of the bases, 5,000, then you can have 4 to the 5,000 different viral genomes arising. And that number goes up with deletions, recombination, and reassortment. It's a huge number. There are only 4 to the 135 atoms in the visible universe. So this is a huge number. So why don't we get new viruses? Why doesn't a flavivirus become something else? Well, there are constraints. A flu virus stays a flu virus. And viral populations maintain these consensus sequences, whereas they could vary extensively to 4 to 5,000. They don't do that. How do you maintain this stability? There are lots of ways, of course. Extreme alterations in the genome simply don't survive selection because DNA can't become RNA or vice versa. You got all the replication proteins to deal with it. mRNA synthesis can't change all that much. RNA structure, codon usage, the physical nature of the capsid with a defined internal space so the genome can't get bigger. And if a virus becomes too lethal, it won't survive. If it's not replicating enough, it won't survive either. So all these properties and more constrain viral evolution. And that's why, despite 4 to 5,000 different possibilities for an RNA virus genome, flu stays flu and polio remains polio. Yet, I'll leave you with some food for thought. In 8 million years of evolution from our ancestors to Homo sapiens, we changed about 2% of our genome. So humans are 98% chimp in terms of similarity of their DNA. By 8 million years, and that's all they did. Poliovirus, in 5 days after ingested to being excreted in the feces, changes 2%, just as much in 5 days. So imagine what a virus can do with 8 million years. It's amazing. Okay, I love that last one, don't you? Ordovicians. Let's go to the last question. Nevitz, Dr. Campbell, the potential for mRNA vaccines to cause cancer is ridiculous. There's no cancer caused by mRNA vaccines. He doesn't know what he's talking about. He spreads misinformation. He's got a lot of followers on YouTube. The Wikipedia says HPV vaccine is only effective till age 21. It's not true. You can get it up to age 45 because previously it wasn't tested in younger people. As soon as it was tested up to 45, it's protective to a certain extent. Not as much as if you take it when you're 12 before you become sexually active, but it still can help. The vaccine is to be given before natural infection. Yes, but there are many serotypes, remember, so you might not have all of them. So that's why vaccination can still be effective after 21. How do males test for HPV? That's a good question. Yeah, there isn't a test for men. Women, of course, you do a pap smear and then you check for viral DNA. But yeah, if a male has a wart, then it would be tested for HPV, a wart or an anal lesion, right? That would be it. Should you get HPV for your daughter who's 22? Absolutely. It's good till 45, Dogma. Definitely. Yes, John cannot go a month without seeing a major media outlet headline saying that COVID got more transmissible because the people don't think about it to the extent that I've just told you about it. It's not about transmission. How many more changes is needed to make it shift compared to drift? Well, of course, shift, you have a completely different HA. So I would say they're 20%, 20 to 30% different in amino acid sequence, quite different. I'm going to stay away from ducks. Yeah, these water birds, they have all kinds of influenza viruses. Ducks and swine are murderous. Glad I don't eat either one. Midge. Hello, Vincent. Welcome. No, there's no need to apologize. It's good. HPV vaccine is good to 45. All right. It's licensed in the U.S. to 45. I don't know what it is in other countries, but it's licensed in the U.S. to 45 years of age. Nevitz, amazing. Glycine to serine change, a tiny difference in a over 400 amino acid residue protein, but that OH group makes all the difference. Yeah, it's amazing, right? Any story starting with European rabbits in 1859 is going to slap. It's a good story, right? Another plague example. Central Park, New York is overcome with Japanese knotweed. Introduced 140 years ago as a pretty plant, it's now outcompeting native plants and is now all around the U.S. Yeah, it happens. New environments, right? We have a creek full of Ordovician fossils, like trilobites. That's right. Yeah, it was a shallow sea in Ohio in the Ordovician. Put that on a T-shirt, which was a shallow sea here in the Ordovician. That would be cool. Riley quotes Martin Luther King Jr., "I'm worried about America." And that was a while ago. Look, RFK Jr. is 98% chimp, 2% brainworm. Oh my gosh. Yep. Oh, you could put 98% chimp on a T-shirt. That's the one. Yeah, that was really good. I like that T-shirt. Dogma, thank you. If we had mesmerizingly good teachers like you, perhaps so many people wouldn't be dunderheads. Science and logic would rule and the world would be safe. Thank you for being here. Well, thank you for saying I'm mesmerizing. That's a good term. I don't know. I'm not sure that. I mean, if every teacher taught like I did in their respective subject, it probably would help, right? But I think I do. My students get taught. They learn virology. Now, of course, they may forget it very quickly. There was someone here before who was a former student. You can ask him. Riley knows someone who's allergic to mosquitoes. What is the future of SARS-CoV-2? Okay, here's the future. Even though you can't predict it. Prediction is hard, especially for the future, Yogi Berra. So I think so this all came from Ralph Baric the first time he was on TWIV. He said, look, we have 4 common cold coronaviruses that cause mild upper respiratory tract infections every year. Nobody really dies from them. Very few people die. They came into people from animals many, many hundreds of years ago, and they probably caused the pandemic because there was no immunity in the population, similar to what happened with SARS-CoV-2. Now, these common cold coronas kept infecting and infecting, and we had a generational shift. The older people died, and they were replaced by young people who aged, but the young people had already been infected with the common cold corona. So as they aged, their infections were not lethal. And eventually we have a population where nearly everyone has immunity to the common cold coronavirus. And then as new kids are born, they get infected, but infection at a young age is milder. It's not zero, but it's milder. Okay, same thing might happen with SARS-CoV-2. Once we have this generational shift, all the older people who don't have good immunity, they're gone and replaced by people who have grown up with SARS-CoV-2. Then it will become a common cold virus number 5. Now, how long does that take? How long does that take? I don't know, maybe 50 years. Ralph Baric said 50 years. So we're 5 years in. It's going to be a while, folks. Yep. Dogma says he got the brainworm in India. No one eats uncooked pork here in the tropics, except your RFK. You can thank us, but even the poor worm died in there. Thank you, Dogma. John, thank you for your support of science communication. Another great week of programming on Microbe TV. Really enjoyed the TWIV. Oh, thank you, John. I was really having a hard time with that second paper. Really, with the names of the viruses, it's really hard to do. So I'm glad you liked it. But there's more than just the papers, right? It's all the other stuff that's a lot of fun too. You'd like to shake RFK's hand? Or why? Why do you want to do that? RFK Jr., you mean? Why do you want to do that? Tell me. Oh, because I have C. diff. I see. Who knows? He probably has poor hygiene. I'm guessing. Okay, COE. Do all positive sense RNA viruses use RNA-dependent RNA polymerase? If their genome can be directly made into viral proteins, what's the pol for? Okay, good question. It's not a noob. It's a great question. So yes, all positive strand RNA viruses have encode RNA-dependent RNA polymerases. Why? Well, as you say, that first RNA gets in the cell, and it's translated into viral proteins. How do you make more RNA? To make a lot of virus particles, you need to have a polymerase that will copy the RNA and make thousands and thousands of copies, right? So that's why you need an RNA polymerase encoded in the viral genome. And even negative strand viruses, which can't be translated, they have an RNA polymerase that they bring in the cell with them. They have to encode an RNA polymerase as well because they have to make a lot of RNA, right? There you go. It's a good question, and it's a teaching moment. It allows me to explain why you need a polymerase. Thank you very much. Questella, goodbye. See you next week. Patricia wants to know if common cold corona is called SARS. Cause SARS. No. SARS is not a common cold. It's a severe acute respiratory syndrome which goes down into your lungs. So the common cold coronas, did they originally? I don't know. It's a good question. When they first spilled over into people, that's a very good question. I don't think we have any data that would address that. But that would suggest that the immunity, population immunity, has also modulated the virulence if, in fact, they caused SARS long ago. I'm not sure that I'm not sure of the classification, but some of them came from, at least one came from a bat, and others came from other animals. Let's see. No. Anyway, we don't know if they caused SARS originally. That's a really good question. I guess they did. RFK Jr. strikes me as the type to skip soap and water after using the toilet. Yes. Do we know which worm it was? It was a pork tapeworm, Taenia solium. He underwent surgery to remove the worm. Pork tapeworm. Now, Dixon told the story of how you get neurocysticercosis, right? Which is what the name is for having this worm in your brain. And I can't remember. It's in one of the TWIPs. But normally these tapeworms, you get a segment. And that's what affects you. But he got eggs. And that's not the right thing to transmit. And therefore, it went to his brain. So we have to go back and look at the TWIP episode from Dixon. But it was Taenia solium. Because, pork tapeworm used to infect many people, but they didn't give them neurocysticercosis. They went into their intestine, right? Older and wiser, SARS-CoV-2 could become a common cold in just 2 to 3 generations. Well, I don't know if it's evolving of the immune system, but rather just widespread population immunity and getting. See, the problem is getting infected at an older age is bad. Even if you are vaccinated, older people don't respond well to infections. And so once those are gone, and then you have people who are infected many times at a young age who age, then they're resistant to the severe disease. So why not 50 years? You replace the older generation in that time. But I just don't know. Right? It's all prediction and theory, hypothesis. Everyone's talking about knotweed now. So that's today's side chat. Personally, Riley wears a mask. I don't think it's a problem to wear a mask. It's your own choice. I wear a mask because I'm over 70 and I don't want to get respiratory infections. So on the train, when I'm in crowded situations, I wear a mask. Trains and subways. But outside, I don't. Although New York City subway sidewalks can get very crowded. But there's a nice wind blowing all the time. How does attenuation reduce virulence? Well, so the attenuation is reduced virulence, right? So in the old days, you would take a virus, like Albert Sabin took poliovirus, and passage it in different hosts. The idea was he selected for viruses that grew well in a different species, and then they would lose the ability to grow well in human cells, and therefore they were attenuated. In my opinion, at least for polio vaccines, attenuation of virulence is all about losing fitness. He made unfit viruses. Albert Sabin made unfit viruses. They replicate poorly, but enough in your intestine to give you immunity. But then there's tremendous selection for increased fitness, which happens within a few days in your gut, and then you end up shedding fully virulent viruses, but by then you've made an immune response and you're protected. So I think it's all about a reduction in fitness, and that can happen in many different ways. For the polio viruses, the main attenuation, the main reduction of fitness mutations are in the 5' non-coding region. Did the 19th century Russian flu become a common cold virus? So that's a good question. I think there's a lot of controversy over whether that was influenza or a coronavirus, right? I think it's not one of the 4 common cold coronaviruses. It's too soon. It's too recent. But if it may be another coronavirus that we just don't know about. I don't think there's enough data to know one way or the other. Barb Mack's great aunt had a tapeworm when she was a child in Africa. She used to tell us about it and laugh. Yes. Many people have tapeworms that can be treated with medication and they will come out, right? Herbert asked, do you know of any other cases besides hepatitis Delta where host Pol2 is used to replicate an RNA template? Yeah, there are a bunch of, let's see. So viroids are circular single-stranded RNAs, and they are copied by host DNA polymerases, which is unusual. And then there are a whole bunch of viruses, viruses like HDV in other species that are also replicated by host RNA polymerase 2. So those are the only example. But, hepatitis Delta was thought to be the only one like that in humans, but now we find them in birds and snakes and all over the world. So there are lots of examples of that. Yeah, so far SARS-CoV-2 isn't becoming milder because there are a lot of elderly people who were not infected as a kid. That's the key. You have to be infected as a kid, and then as you age, you have good immunity against severe disease. So there are just too many people around today that were around in 2000. And until they're gone, it's still going to cause a lot of disease. Yeah. Oh, this is interesting. Miasma Fresh Spray. Good name for it. Good name for it. Okay, folks. Looks like you are exhausted tonight. Let's see. We had 200 people here at one point. And we have a good ratio of likes to concurrent viewers. So I want to thank you all for that. Let me thank our moderators tonight. We had Barb Mack. Wow, Barb Mack, it's pretty late for you. Thank you for staying. Really appreciate it. Vanity Nutrition. We had Steph SF. We had Tom. And we had Les. Thank you all for moderating. I appreciate your work here. Tireless, but I appreciate it as always. And I want to thank everybody for coming with your great questions and side tracks, which are always amusing. Today was the peace sign and then some kind of invasive plant. So when are we going to be here next? The 18th of June. At 8 p.m., we'll have office hours. That's 8 p.m. Eastern. And, of course, the same day, 1 p.m., we are going to have TWEVO 115 live stream. So you might want to check that out as well. Noir, thank you very much. Microbe TV is the best thing on YouTube. I don't know. There's some music channels that are really good, but I appreciate the thought. Maybe it's really good for science. I really appreciate that. All right, folks. It's been a blast. It's been a slice. I hope you are all safe. Love you all. See you next week. Okay. Stay healthy. Bye-bye.